Good morning, everyone. Well, at least it's morning for me. So, final lecture on this little portion of the class. And we're going to talk about bioinformatics today, which was not a thing that was a thing when I was in graduate school, really. It is relatively new. It's also an extremely lucrative field. If any of you are inclined in both the realms of mathematics / computers and biology, there are lots of jobs to be had in the bioinformatics field, because oddly enough, those are skill sets that don't overlap very commonly. People that are good at coding and math, and people that understand biology. So let's talk about bioinformatics and how it works. So essentially, when we're talking about bioinformatics, it's a very broad field. It essentially means using your computer and lots of different suites of programs to study biology. And as you can sort of get the hang of now we've learned more and more about how genetics works, and you guys understand things like sequence-specific transcription factor binding and things like that. It should become obvious to you that computation and computational analysis of genetic sequences can be very powerful, and indeed is very powerful. So, most of us as geneticists begin our bioinformatic experience with some kind of data. That data set can be something like the entire genome, or it can be something simple like this. So this is a DNA sequence of a gene called Pygopus from fruit flies. This is actually the CDNA sequence. So this is a piece of mRNA for this gene that has been basically copied back into DNA. So that process removes any of the introns that were present, right? Because they get spliced out to make the mRNA. And then if you copy the mRNA back to cDNA, you just copy the message. So that's what this is. It's about 2,500 bases long. And why is this interesting to me? Why am I showing it to you? Because this was my first time using any kind of bioinformatic software, because this is the gene that I studied for a lot of my PhD. So this title is the title of the first paper from my PhD, studying this particular gene. And I wanted to use this as a tool to show you some of the stuff you can do with bioinformatics. So with bioinformatic programs, you can use them to do a lot of different things and answer a lot of different questions, such as, does this sequence that I'm interested in contain a gene or not? Does this sequence that I'm interested in contain functional DNA elements like promoters, splice sites, enhancers, those kinds of things? Does this sequence encode any kind of polypeptide? If so, what is the sequence of that polypeptide? Because we can translate a DNA sequence or an RNA sequence very easily using computers. Does the sequence predict any potential structural features for the protein that it's going to make? And we can often do that by comparing polypeptide sequences between different genes and different species. Going on from that, we can ask if the DNA sequence that we have, is it homologous or similar to sequences from any other genes, both within that same species and amongst other species too. Using that kind of information, we can actually look at evolutionary relationships between species using bioinformatics. Because essentially, right, if I have the gene for RNA polymerase, which I do, and presumably this guy, who is also with me again, he has a gene for RNA polymerase, right? He has the cat version of that same gene. We both need it because we're biological organisms, an RNA polymerase is required for both of us to live. But we diverged a long time ago on the evolutionary map. So our related genes probably have accumulated several different mutations over that time. And you can use differences between conserved genes to see how closely related species are. Thanks, Max. So let's look at a very simple example of what computer programs can do. So I'm going to take that sequence that I studied, that gene that I studied as a PhD student. It was a gene that we discovered in a genetic screen. So we knew it was important for something because if we knocked it out, it had an interesting phenotype. And then we wanted to know what it did. So the first thing you want to do is figure out, well, what protein does this gene actually make? And that's pretty easy to do because we could take the CDNA sequence that I showed you earlier, and we can run it through a translation program, which essentially does exactly what a ribosome does. It's going to take that sequence and figure out what amino acids that sequence codes for. But when you give a computer a piece of DNA or RNA sequence, you have to tell it where to start translating. And that's because, remember, there are different reading frames in a genetic sequence because of the triplet nature of the genetic code. So what I mean by that is this, right? Here is a very short DNA sequence. It's the first 10 bases of the Pygopus cDNA. The first reading frame for this piece of genetic information starts at the very end, right? So those first 3 bases, A, T, G, code for methionine. The next 3 bases, A, C, C, code for threonine. And the next 3 bases after that, C, A, C, code for histidine. This is called the first reading frame because it begins at the first base. For this piece of DNA, the second reading frame would begin at the second base. And that would be a first codon of TGA, because we're now starting at base number 2. And what you can see here is if we start at the second base using the second reading frame, now the amino acids are different, right? We have a stop codon, actually, is the very first codon, TGA, and then a codon that encodes for proline, and then threonine. And if we move to the third reading frame, starting at the third base, again, we now have 3 different amino acids encoded for. So what most translation programs will do is give you all 3 of those reading frames so you can see which one might be correct if you don't already know the answer to that. So if I take this sequence, this cDNA sequence of the Pygopus gene, and I stick it into a translation program, this is the kind of output that I get. A lot of these are available easily on the internet. So there are 3 different reading frames here shown in each of the boxes, frame 1, frame 2, frame 3. There's actually technically 6 different reading frames because we could look at this sequence in the opposite direction. At the moment, we're looking at it 5' to 3', but we could switch it around and look at the 5' to 3' from the opposite direction. There would be 3 more reading frames if we did that. In that case, we would start right at the bottom here with that last 3 bases TGA, but we would be reading it from the other direction, AGT. I don't need to do that because I know the orientation of this piece of DNA. But what you see when you look at these 3 reading frames is that what these programs tend to do is highlight certain important things. So open reading frames are highlighted in red here. What is an open reading frame? It is simply a portion of the genetic material that starts at a start codon, methionine, and ends at a stop codon. That's called an open reading frame because it means that in theory, a ribosome could start and just read that sequence until it reaches a stop codon. And what you actually see if you look at these 3 reading frames is that frame 1, there's a big open reading frame where a ribosome could read the entire DNA sequence and put in different amino acids for each codon without reaching a stop codon. By the way, the amino acids here are written in a single letter code. So an M is methionine, a T is threonine, and an H is histidine, for example. So if you look at the first reading frame here, met, threonine, his. We go back to the reading frame 1, you see it's MTH, which is those same 3 amino acids. Reading frame number 2, if you start at that second position, we know from looking at this that it was a stop codon, a proline, and a threonine. So if we look at the second reading frame, we see a little dash that represents a stop codon, and then a proline and a threonine. The third reading frame itself is, there aren't any open reading frames at all in that one. So it's obviously way quicker for a bioinformatics program to translate this piece of DNA than it would be for me to sit down and figure out every individual codon myself. So that's just one example of what bioinformatics can do. That's very simple. You can just translate a piece of DNA. You can also plug in much longer pieces of DNA, for example. You can do a whole genome. And there are plenty of short sequence elements that a computer program can show, that can figure out where they are. So you can look for things like promoters, for example. Response elements, those are enhancers, right? Like the glucocorticoid response elements that we looked at previously. You could pick out start codons, stop codons, splice sites are known, polyadenylation signals are known, and there's plenty of other sequence elements that these programs can pick out for you, obviously in a much quicker way than you can do using the human eye. One particularly important aspect of bioinformatic programs is their ability to predict where a gene might be. And that's really important, right? I can then give a program, for example, the entire human genome, and it can predict genes that are in that genetic sequence. And computer programs can predict protein encoding genes, mostly by looking for open reading frames, and also genes for very important non-coding RNAs as well. And they kind of do this in different ways. So they can look for things like start and stop codons and splice sites. That's looking for different signals. Or they can search by content for things like codons, because it's very unlikely that you'll get a big open reading frame that will occur by chance. So that's how we actually got to this. When the human genome was first sequenced, that's how we got to this number of about 22,000 genes in the human genome. It was by doing gene prediction using computers. So looking at ancestral genes, in other words, comparing things like RNA polymerase between me and my cat, that can help us find genes as well, because it's often true, because of the way evolution works, right? Genes don't just pop up out of nowhere. Things change slowly over time. So if you find a gene in one organism, you can look for it in another organism by comparing sequences. Here's an example of that. This is the LAC-Y gene in E. coli versus a different bacteria. And you can see that these are relatively closely related, so that 78% of the bases in these genes are a perfect match. Here's just a small example of the gene. Anything that's in red does not match. Anything that's in black does match. And the first line is one bacterial species, and the one below it is a second bacterial species. So this is another way you can look for genes in closely related organisms. Here's another example of that I wanted to bring up, which is something that we all care about at the moment. So what you're looking at here is a diagram of the entire genome of SARS-CoV-2, the virus that causes COVID-19. It was very quickly sequenced. So we have the entire sequence. You can see how long it is. I can't, my video's in the way. Let's see if I can move my video. Yeah, so it's 29,903 bases long. And this little map shows you different things that are interesting in that genome, one of which is the spike protein, which is shown around the 22,000 to 24,000 base pair mark. And then what you're looking at in the long pink line, second from the top, is basically that what is it? These are amino acids now. So this is 1,285 amino acids that make up this spike protein. And at the very bottom here, panels A and B, it shows you the amino acids and how the current virus, human SARS-CoV-2 at the top there, is conserved with the amino acids of other versions of this coronavirus. In bats, pangolins, human SARS-CoV is the original SARS virus, and then a couple of the ones in bats. Why is the spike protein so important to look at? Well, it's basically because of what the spike protein actually does for this virus. So in the bottom right-hand side of this slide, you can see a picture of the coronavirus, which you guys have probably seen now many times, some version of that. And it's got these red things on the outside. That's why it's called a coronavirus, because those are like its crown or corona. And that's the spike protein. And the reason the spike protein is important, as you can see in this cartoon, is that there is a human host cell on the left with a protein sticking out from the cell membrane known as the ACE2 receptor. That is how this virus gets into human cells, how it infects us. And the protein spike, part of this corona or crown of proteins, interacts with the ACE2 receptor. And that's what that recognition event allows this virus to get into human cells. So this spike protein that we're looking at here is really important. And what you can see if you look at the amino acid sequence and you compare the human SARS to these other different versions, sorry, the human CoV-2 virus, the current virus that we're suffering through. If you look at the amino acids and you look at how they compare between other versions of this virus, you see there are some differences. The most obvious one is the polybasic cleavage site at the top there. And then there are some other ones shown in different boxes. And essentially what this shows is that through mutations, these other viruses are changed in their spike protein, which allows it to be much more infectious on human cells. And what this paper, which was just published in Nature, concludes is, said at the bottom here, analyses clearly show that the current virus, CoV-2, is not a laboratory construct or a purposely manipulated virus. How do they know that? They know that because, and I want to cut to the chase here, what's in bold, this spike protein, again, a portion of it is shown at the top here, it binds to the human ACE2 in a very tight way, but there is evidence that it is not, that was not done by humans, because the changes that were made are not obvious and were most likely to have arisen by natural selection. So that's at least the most likely origin of this virus. So it's unlikely that this was manipulated in some Chinese lab. It could have escaped from a Chinese lab that was studying coronaviruses, but it looks like it was not man-made from sort of the bottom up. So when you do compare genes between different species, if you find two genes that do the same thing, like my version of RNA polymerase and my cat's version of RNA polymerase, those will be called orthologs, which is how you say the same gene in a different species. If within that species you contain two different versions of the same gene that do two different things, those types of genes, in other words, genes of a gene family, are called paralogs. And those are just two important terms that we use when we are comparing genes from both within and between species. As the last probably 15 years have gone by, and we've got extremely good at generating a lot of DNA sequencing data on the cheap. The amount of genetic information available to researchers has just exploded. It's crazy. And there are now many databases that are out there that scientists use to search all of that information. And there are several of these databases but there are a couple that are very typically used by most people. And this just gives you some idea of what most of those really important computer databases are. There are several for nucleotide sequences. GenBank, the US one, is used by many, many, many people worldwide. There are databases that contain just amino acid sequences. The SwissProt one, shown in the second category here, is the most famous one. And there are other ones that contain things like where you can search for different protein motifs and things like that. And then calling back to the gene expression stuff we were discussing in the last lecture through DNA microarrays and RNA-seq, there are gene expression databases too. And these ones shown here are all free to use. So that's an amazing resource for genetic researchers throughout the world. It's particularly important because if you discover a new gene and you don't know what it does, you can search for genes that look like it in other animals. And maybe there's some homology that can show you what it might be doing. And that's really powerful. And indeed, when I discovered the Pygopus gene, that's the first thing that we did in my lab. There's a very famous tool called a BLAST tool, stands for Basic Local Alignment Search Tool, which basically allows you to upload any DNA sequence you want and compare it to every other DNA sequence that has ever been discovered. And if it's similar to one of those sequences, it'll tell you. This table essentially is an annotated result from a BLAST program. I was going to show you the version that you get if you BLAST the Pygopus sequence, but I didn't because it's too complicated in sort of the output form. But this is basically what it tells you. It gives you a string of matches based on the most similar first to the least similar last, and you get hundreds of thousands of matches sometimes. This just shows you the top 10. And so this was a BLAST search that was done using a certain sequence. Phenylalanine hydroxylase is the gene. And this person was looking at the human version of the gene. So if you upload a human version of the gene and you do a search against everything, the top match is always going to be the thing that you began searching, right? So if you search with the human gene, you're going to get the top hit as the human gene, and that matches 100% of the amino acids. But you can see that this gene is present in all of these other species: orangutan, mouse, rat, chicken, xenopus, zebrafish, all these things. And the E value on the right tells you how similar it is, and it also tells you the percentage of identical amino acids. And so you can see as you get further away from humans down the evolutionary tree, the percentage of identical amino acids goes down because of evolution. So if you go back to sort of nematodes or worms, since we diverged from nematodes, our genes have been mutating, changing the number of amino acids that match between us and worms or fruit flies. So you can see, you can look for different genes in different species based on your sequence. And sometimes that's really useful because sometimes there are certain, as you guys know, right, proteins have domains or motifs in them that do certain things. So remember that if you're a transcription factor, you can have a DNA binding motif or domain, like a zinc finger domain. And you can also have some kind of transcriptional activation domain. And those two domains, which are part of the same protein, basically contribute to the jobs that that protein does. So you could do that with proteins you're interested in to maybe get some clues about what they're doing. And so when I did this with the Pygopus protein, when we discovered it, I looked at it in some databases, and there were some predicted domains. The top line here tells you that at positions 39 to 45 in this protein, there is a nuclear localization signal, suggesting that this protein might reside in the nucleus. And what you're looking at in those three panels is some pictures I took that are in that first paper that show that this is true. So in red is an antibody that basically shows the nucleus. In green is the Pigo protein. And if you merge them together, you see that the Pigo protein is absolutely inside the nucleus. And I showed that through some work that's not shown here, that if you mutate those nuclear localization signal residues, it is no longer confined to the nucleus. So that was useful to know. And there's another domain in this protein right at the N-terminus. So if you look at the regions here and look at the feature key on the left, it says that there is a zinc finger motif. And actually, the description of this particular feature is that it is a PhD type domain, which I thought at the time was hilarious because I was doing my PhD. That's the kind of thing that I think is funny. But what that actually means is plant homeodomain, and it's a zinc finger domain that's in positions 747 to 805, right at the end of this particular protein, that suggests that its molecular function is, as you can see at the bottom of this slide, a transcription factor of some kind, and indeed that's what Pygopus does. So this kind of homology searching can be really useful when you've identified a gene but you don't know what it does, because from using these databases, I was able to predict that this is a nuclear protein that's probably a transcription factor. And that's because this zinc finger PhD type domain had been identified in other proteins, and a very similar version of it was present in this one too. Okay, so that's just scratched the surface of bioinformatics and the kind of things that you can do as a geneticist these days by just sitting at your computer. So it's kind of amazing. All right, you guys, I hope you're doing all right. One lecture, one topic left to cover. Take it easy. Bye-bye.